Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Remember that initial feeling, that surge of hope when monoclonal antibodies first appeared, like a powerful new weapon against viruses, especially SARS-CoV-2.
Oh, absolutely. The idea of engineering these super precise tools to just, target and neutralize a virus was huge.
And they did work, for a while anyway. But then, well, the virus started doing what viruses do.
It started evolving, finding ways around these very specific defenses we built.
Yeah, it was almost uncanny, wasn't it? How it could just pinpoint the weakness, the Achilles heel of a single engineered antibody.
It really drove home that we're in this constant dynamic biological arms race.
We are, and understanding how the virus actually manages that, how it finds and exploits those weaknesses, well, that's critical if we want to stay ahead.
Exactly, which is precisely what we're diving into today.
We're looking at some really interesting new research from TWINCORE and collaborators in Hanover and Bern.
They've taken a really close look at the nuts and bolts of this viral escape, how it happens at a molecular level.
And they're proposing, well, a smarter strategy moving forward, basically borrowing a page from nature's own playbook.
Yeah, so our goal here is to unpack what they found, get a handle on the mechanics of how the virus gets away, and, see what it all means for fighting these rapidly changing viruses.
Okay, let's do it. Maybe first, just a quick refresher on antibodies generally. They're fundamental to our immune system, right? They find invaders, viruses, bacteria.
And neutralize them, yeah. Or flag them for destruction. Think of them like very specific keys. Each antibody key is shaped to fit a particular lock on the surface of a pathogen, like a protein on a virus.
And when that key fits the lock,
it can either block the virus, maybe stop it from entering our cells, or it acts like a signal flare, calling in other parts of the immune system to deal with it.
And naturally, our bodies don't just make one key. It's more like a whole key ring, right? A mix.
That's the polyclonal response. Poly, meaning many. Lots of different immune cells, making a diverse range of antibodies that recognize multiple different locks on the virus.
It gives you options. Redundancy.
Exactly. If the virus changes one lock, maybe another key on the key ring still fits. But the treatments we developed, those were often monoclonal antibodies.
Mono meaning one.
Right, lab engineered. Millions of identical copies of just one specific key designed to hit one precise spot on the virus. These were used as medications.
And they were effective, certainly, for many people initially. They could block the virus. But that single key approach, that's where the weakness lies. Dr. Matthias Brun, one of the lead authors here, highlighted this.
Yeah, he pointed out that if you're relying on just that one type of key, the virus only needs to change that one specific lock it targets.
Just a tiny change, a single mutation.
A single mutation in the virus's genetic code can sometimes be enough for that specific antibody key to not fit anymore. And then it just loses its effectiveness.
And viruses are mutation machines. They're constantly making small errors when they copy themselves.
And if your treatment puts pressure on just one spot, any random mutation that happens to change that spot gives that virus a huge advantage. It escapes the antibody, replicates, and takes over.
Which brings us right back to this study. The researchers really wanted to get into the weeds, understand the fine details of how this escape happens.
How, where, and crucially, use that knowledge to figure out better ways to treat these infections, ways to counteract this escape.
So how did they approach it? It seems like they tackled it from a few angles.
Yeah, a really smart, multi-pronged approach. First, they use hamsters, an animal model.
Okay.
They gave these hamsters some potent monoclonal antibodies known to neutralize SARS-CoV-2, and then they infected them.
So they watched to see if the antibodies would protect them.
Exactly. And, well, it wasn't perfect protection.
So even with the antibody treatment, some hamsters still got sick.
Some did, yes. They had what the researchers called breakthrough infections.
Okay, and what did that tell them?
Well, when they took virus samples from those sick hamsters and looked at the genetic sequence...
They found changes.
Specific changes, yes. In one instance, they found a particular mutation, G485D they called it.
Yeah.
This is really strong evidence. It suggested the virus hadn't just slipped through, it had actually mutated inside the hamster, specifically to get around the antibody treatment it was facing.
Wow, so it evolved during the infection in response to the treatment.
Precisely, showing that selective pressure in a living system.
Okay, that's compelling. But animal studies can be slow, right?
They can be. So To study the process more rapidly and, under more controlled conditions, they also developed an in vitro system, basically, using cells in a dish.
A lab assay.
Right. They used a clever trick, a modified virus. It was a vesicular stomatitis virus, but they engineered it to wear the SARS-CoV-2 spike protein on its surface. That's the main target for many antibodies.
So it looked like SARS-CoV-2 to the antibody, but maybe easier to work with in the lab?
Exactly. This let them quickly apply pressure with a specific antibody in cell culture and watch, almost in real time, for any viral variants that mutated to escape that antibody's effects.
A faster way to see escape happen. And did it confirm the hamster findings?
It did. The lab system successfully recreated this escape scenario. And Maureen O'Barra, a PhD student on the project, noted something really key here.
What was that?
The location of the mutation that allowed escape was directly linked to the specific antibody they used. The changes consistently popped up right where that particular antibody was supposed to bind on the spike protein.
So the antibody itself was guiding where the virus mutated, like pressing on one specific spot until it changed?
Very much so. Targeted evolution under pressure.
And they confirmed these specific changes using genetic sequencing.
Yes, detailed genetic analysis. They sequenced the virus genomes from the hamsters and from these lab-based escape experiments to pinpoint exactly where the changes were happening.
Okay, so they looked at the virus. Did they also look closely at the antibodies themselves?
Oh yeah, definitely. In-depth antibody analysis. They used techniques to measure things like binding affinity, basically. How tightly does the antibody stick to the spike protein?
Like the strength of the key in the lock.
Good analogy.
Yeah.
And also epitope mapping, which is figuring out exactly where on the spike protein the antibody binds, mapping the specific lock location.
Got it. And you mentioned other immune functions earlier.
Right. This is really interesting. They didn't just look at neutralization, simply blocking the virus. They also ran immune function assays.
Meaning?
Testing if these antibodies could activate other parts of the immune response. Could they, say, help immune cells eat the virus? That's phagocytosis.
Okay.
Or could they trigger other processes to clear out infected cells? They looked at activating specific immune cells, like neutrophils or natural killer cells.
So seeing if the antibody could do more than just block entry.
Exactly. Could it act as that signal flare we talked about?
Okay, so quite a comprehensive approach. They saw escape in animals, recreated it in the lab, pinpointed the mutations, analyzed the antibody binding. What were the big takeaways? How does this help us fight back?
Well, first, seeing those specific single mutations causing breakthrough infections in the hamsters was a big one. It showed viral escape isn't just theoretical or something that happens slowly over populations. It can happen relatively quickly, under treatment pressure, within a single infected individual.
That's a bit sobering.
It is. And the lab system confirmed how targeted this escape is, happening right where the antibody binds, just as Maureen O'Barra observed. The virus finds the path of least resistance.
Right, but then they looked at the antibodies themselves and this natural process. What was it called again?
Somatic hypermutation, SHM. It's fascinating. It's how our own B cells naturally refine the antibodies they produce over time.
Like editing or improving the keys.
Kind of, yeah. Making tiny changes, often improving how well they bind, how tightly they stick to their target. It's nature's own antibody optimization process.
So they compared antibodies that were sort of raw, less refined, versus ones that had gone through this SHM process.
Exactly. They compared germline antibodies closer to the original genetic blueprint with hypermutated ones that had undergone significant refinement. And they found something really interesting.
Which was?
The amount of this refinement, this hypermutation, actually influenced where the virus tended to mutate to escape.
Huh. So a more refined antibody could change the escape route.
It seemed to shift the hotspots for escape mutations on the spike protein in some cases. And these more refined hypermutated antibodies often showed much, much better binding affinity.
They stuck tighter.
Way tighter. And often worked better against different versions of the virus, like the alpha and delta variants that emerged earlier.
And they saw this improvement in the real world too, right? With people who'd had hybrid immunity.
Yes, exactly. They compared antibodies from people who were just infected versus those who were infected and then vaccinated. That vaccination boost significantly increased the binding strength, the affinity of some antibodies.
Made them stickier better.
And it even broadened their reactivity, making them better able to recognize newer variants, like Omicron.
So this natural process, somatic hypermutation, makes antibodies better and more adaptable.
It's nature building a robust defense. And that's exactly where the researchers got their big idea for a better strategy. Dr. Brune literally said they were copying the recipe directly from nature.
Don't use a single key.
Use the whole keyring. Use combinations of antibodies.
Right.
Mimic that diverse, natural polyclonal response.
So find antibodies that hit different spots on the virus, different epitopes?
Precisely. Identify antibodies that bind to distinct, non-overlapping locations on the spike protein. So changing one spot wouldn't affect the other antibody.
And then combine them in a treatment, a cocktail.
Yes. And when they tried this in their lab escape assay, combining just 2 different neutralizing antibodies that targeted separate sites, the results were striking.
What happened?
They completely shut down the emergence of escape mutations. It just didn't happen in their system.
Wow. Just 2 was enough.
In this setup, yes. It shows that making the virus need multiple, simultaneous, specific mutations to escape changing 2 different locks at the same time makes it vastly harder, statistically much less likely.
That makes intuitive sense. Much harder target to hit.
Definitely. And they explored one other twist, too. Adding a non-neutralizing antibody to the mix.
An antibody that sticks to the virus but doesn't actually stop it from entering cells on its own? What's the point of that?
Well, even though it didn't neutralize by itself, when they combined it with a neutralizing antibody, this non-neutralizing one seemed to boost other immune functions.
Like the cell eating, the phagocytosis.
Specifically, it enhanced the ability of immune cells called neutrophils to gobble up the virus, and it also boosted the activity of natural killer cells, which are important for killing infected cells.
So it's like it helps rally the other troops, even if it's not blocking the door itself.
Kind of, yeah. It suggests a cocktail could attack on multiple fronts, block entry, and ramp up clearance by other immune cells.
Okay, so pulling this together, the implications seem pretty clear. Using just one monoclonal antibody against a virus like SARS-CoV-2 is inherently risky.
Very risky. This study really drives home that viral escape isn't just a lab curiosity. It's a real, practical problem that can emerge rapidly during treatment.
But the good news is the solution seems clear too. Use combinations.
Absolutely. The evidence strongly supports using antibody cocktails targeting multiple different sites on the virus. That significantly stacks the odds against the virus escaping.
That feels like a crucial lesson for developing future antibody therapies, not just for COVID, but maybe for flu, HIV, other viruses that change.
Potentially, yes. Any rapidly evolving viral target.
And thinking beyond just blocking the virus seems important too, incorporating antibodies that might boost other immune functions.
Yeah, those non-neutralizing but functionally active antibodies. Adding them to cocktails could provide another layer of defense, especially maybe for treating people who are already sick, to help clear the infection faster.
It's really about harnessing more of the immune system's toolkit.
Exactly.
Yeah.
And fundamentally, it's about learning from nature. Understanding how somatic hypermutation works, how B cells diversify antibodies. That process can directly inform how we design better therapeutics, maybe even antibodies that anticipate future viral moves.
It's like bio-inspired engineering for medicine.
Pretty much. And Professor Ulrich Kalinke, who was involved, really emphasized the collaborative aspect too. Bringing together different expertise from different institutions was key.
These big biological puzzles usually require teamwork.
Almost always.
Now, we should also touch on the limitations, right? Every study has them.
Of course. And the authors were upfront about these. For instance, fully reconstructing the original germline sequence of some antibody regions, especially complex ones like the CDR3 loop that's vital for binding, is technically tricky.
So some conclusions about the effects of that natural refinement process, the SHM, are maybe more solid for other parts of the antibody.
That's a fair way to put it. Also, tiny technical things, like even with careful purification, antibodies might slowly clump together a tiny bit in the test tube, which could potentially slightly influence the very precise binding measurements.
Okay. Lab realities.
Lab realities. And in that escape assay where they saw mutations pop up, sometimes multiple mutations appeared together.
Yeah.
It's hard to be absolutely certain if they all happened in the same virus particle or just coexisted in the virus population, the quasi-species.
Right. And escape itself has an element of chance.
It's stochastic, yeah. Repeat the experiment, you might get slightly different results just due to random chance.
And that finding about combining neutralizing and non-neutralizing antibodies, boosting other immune functions, that needs more testing.
Definitely. That was based on the lab data, the in vitro assays.
Yeah.
A really promising lead, but it needs confirmation in animal models to see if that cooperative effect really translates to a living system in vivo.
Okay, important caveats. So bringing this back to the listener, what's the takeaway from this deep dive?
I think it really shows you the incredible level of detail scientists are working at to understand this constant battle, this arms race between our bodies, our medicines, and these ever-changing viruses.
Yeah, it highlights why making treatments that work long-term isn't straightforward when your target keeps moving.
Exactly. But it also shows how researchers are finding inspiration in the elegance and resilience of our own immune systems.
Looking at nature's solutions.
Nature's had millions of years to figure this stuff out. We can learn a lot by looking closely.
So to wrap up, the core message seems to be, against a shapeshifter virus, a single magic bullet is vulnerable.
Right. The path forward involves combining forces, creating multi-pronged attacks, building treatments that are diverse and adaptable.
Much like our own natural immune response tries to be.
Precisely. Make the virus overcome multiple hurdles at once.
It really does make you think, though, beyond just viruses and antibodies, this idea of complexity and redundancy in biology. Things that might look messy or inefficient. Maybe they actually hold clues for designing better, more resilient technologies in general, whether it's medicines or new materials or even complex human systems like supply chains. Could mimicking nature's layered, adaptable strategies be a key to building more resilience everywhere?
That's a fascinating thought to leave people with. Yeah. Where else can we learn from biological resilience?
Thanks for tuning in to the Health Headliner podcast by Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.